we're back with the digestive system, so let's continue. The mucus cells produce bicarbonate-rich mucus to protect stomach wall from acid. We answered that, so we can go with this. Still, let's go over this here. So we have surface mucus cells. They secrete mucus. We have mucus neck cells. They are lower in the gastric pit. This is called the lamina propria. These are gastric glands. We have parietal cells. If you remember, they secrete HCl, hydrochloric acid, and intrinsic factors. We have chief cell. So we know that the chief cell secretes pepsinogens and gastric lipase. We also have G cells, which secrete gastrins, and gastrin increases gastric motility. So, there's something here. The digestion of proteins, role of proteolytic enzymes. Proteolytic enzymes, also called proteases, they break down dietary proteins into their constituent amino acids. They are produced by 3 different organs: the stomach, the pancreas, and the small intestine. So now we get to know that there are 3 places that peptides, proteins are digested. So one of them is the stomach, as we know here. The other one is in pancreas, and also the small intestine. So when food enters stomach as a protein, the gastric mucosa are stimulated. Then they cause the gastrin to be released. We see here we have G cells, right? And because of gastrin, chief cells are also stimulated, so they release pepsinogen. The parietal cells also are stimulated, so they secrete hydrochloric acid. So hydrochloric acid denatures the proteins. Pepsinogen that is released has an enzymatic action of pepsin. So there's more pepsin, and then the pepsin causes the hydrolyzation of peptide bonds on the amino terminal side. So the peptide bonds are hydrolyzed via the amino terminal side. Now smaller peptides are transferred to the intestine. This is where we have acidic contents, so the pH is low here, and this is what happens inside the intestine. So we have the release of secretin. So pancreas is stimulated by secretin, which releases bicarbonate, and the HCl is neutralized with bicarbonate that is resulted by secretin, and the pH is raised to 7. The release of so we expect that the pH of the stomach is 2, as we know, and then it increases back to 7 in the small intestine. Now there is a release of cholecystokinin because of the smaller peptides. So cholecystokinin causes the secretion of trypsinogen, chymotrypsinogen, procarboxy peptidases A and B. And trypsinogen is turned into trypsin with enteropeptidase, and trypsin causes these two to turn into chymotrypsin and carboxypeptidases A and B instead of procarboxy, and these go in a loop to hydrolyze the protein efficiently, and then the carboxypeptidases A and B turn into carboxyl terminal residues that are removed. This is a degradation of smaller peptides, and aminopeptidase is also turned into used to hydrolyze amino terminal residues, and then we have free amino acids that are absorbed into the blood and eventually in our liver. That's why if we digest too much protein, then we will have an enormous storage of proteins in our liver, and that can damage the liver, right? Cool. The stomach has the following 4 parts. Again, it's the there's the body, there is the antrum, I believe that's the name. There is the pylorus, that is the end part, and there is I forgot fundus. So yeah, it was antrum, fundus. These names are tricky to memorize, to know. So we should kind of start memorizing with, like, there is funding that comes to our stomach, and then the body is basically the body of the stomach, where the funding is processed by a body actually, and then antrum is a bunch of ants having rum. Man, it's horrible. And pylorus is we know there's the pyloric sphincter, which should do the job, but still yeah, so fundus. So fundus, we have the body, there's an organizing body that processes the money, the funding, and then we have ants drinking rum, antrum, and pyloric sphincter, which is the ant, so pylorus. Chief cells secrete pepsinogen and another enzyme, so gastric lipase. So we can imagine our chief officers, the sheriffs, having fat belly, so they have gastric lipase too, and pepsinogen because they're drinking Pepsis, right? The parietal cells secrete HCl, hydrochloric acid, and the intrinsic factors, right? So intrinsic factor is needed for vitamin B12 absorption. I don't think we had it here, so here. Intrinsic factor is for vitamin B12 absorption. Cool. Okay. I still got the intrinsic factor, but I didn't notice it was for vitamin B12. So G cells secrete gastrin, and it's a peptide. Gastrin is a peptide hormone that increases HCl secretion and gastric motility. So yeah, we can forego with that. should know it not only increases gastric motility but also hydrochloric acid secretion. After processing in the stomach, food particles are now called bolus, chyme. So bolus is what the food is turned into in our mouth, but after processing the food in the stomach, so after the stomach, it is now called, the food particles are called chyme. So chyme is mixed food. The direction of food is from the fundus. So we have lower esophageal sphincter that opens. Now fundus is empty here, and we have the chyme. The chyme goes passes through the pyloric sphincter closed. And here we have the duodenum, and this is the direction of food again. Chyme exits through pyloric sphincter and enters the duodenum. Perfect. The stomach has a pH of about 2. Yes. So the esophagus has 5 to 7. Stomach is 2 to 5. Duodenum is 6.8, a bit more acidic. pH equals 5 to 7. Yes, 5 to 7. Now for duodenum is 6.8. So here we have duodenum 6.8. So we have to make sure, is the pH of duodenum 6.8 or 8.5? I mean, it's also tricky because duodenum has to kind of be 8.5, more basic, to neutralize the acid that's coming from the stomach. So yeah. What is the first part of small intestine in the site of the majority of chemical digestion? Is duodenum, right? And then we have the jejunum and ileum. This is same. What is the pixelized here? Duodenum, deodorant, macromolecule breakdown, wood chipper, small intestine, rubber hose. And who came up with these? What is it going on here? Okay, I get the deodorants being duodenum, but like the macromolecule breakdown, like digestion, the majority of chemical digestion, because like cutting down wood is like a physical digestion, not a chemical one. Yeah, it's crazy. And the small intestine trying to say that it's related to duodenum, man, whatever. Okay. The following enzymes are found in the so that we have disaccharides, aminopeptidase and dipeptidase, and enterokinase. These should be found in duodenum. Yeah, so can put this hard. What enzyme is a brush-border enzyme that breaks down maltose, isomaltose? This should be disaccharidase. Disaccharidase, yes. So maltose is, we have maltase for maltose. Reactions catalyzed by disaccharadases. So maltose is glucose plus glucose. sucrose isomaltose is 3-glucose plus fructose. I think by 3 here it means I don't know, something refers to the 3 isotope of glucose. And then lactose is glucose plus galactose. So yeah, remembering that so MGG, SGF, and LGG LGLG. So LGLG gives you the galactose and glucose, right? And maltose is again glucose and glucose, so it's GG. And sucrose is glucose and fructose, SGF, standard girlfriend. So we need a mnemonic for, we have MGG, maltose equals glucose plus glucose. We have SGF, sucrose equals glucose plus fructose, and LGLG, which equals lactose equals galactose plus glucose. Yeah, that should do the job. What is a brush border enzyme that breaks apart amino acids? It should be pepsin and aminopeptidase. Okay, no way we got that. So we have amino acids of protein fragments, brush border enzymes, apical membrane. Proteins and protein fragments are digested to amino acids by pancreatic proteases, trypsin, chymotrypsin, and carboxypeptidase, and by brush border enzymes, carboxypeptidase, aminopeptidase, and dipeptidase of mucosal cells. The amino acids are then absorbed by active transport into the absorptive cells and move to their opposite side, so transcytosis. And the amino acids leave the villus epithelial cell by facilitated diffusion and enter the capillary via intercellular clefts. So we have lumen of intestine. We have pancreatic proteases. We have absorptive epithelial cell. We have amino acid carrier. We have capillaries, and the active transport is from here, and the passive transport is from here. So yeah, and here we have procarboxypeptidase, chymotrypsinogen, pancreatic juice. This pancreatic juice is something else, and trypsin, carboxypeptidase, chymotrypsin, peptidase, Peptides are turned into amino acids, di and tripeptides. We have peptidases, and they turn into amino acids. We have enterokinase as well, which turns trypsinogen into trypsin. What is a brush border enzyme that activates trypsinogen to make trypsin? We just read enterokinase, also called enteropeptidase. So what was the thing before? Was it aminopeptidase? So there is a difference between aminopeptidase and enteropeptidase. Be careful, as enteropeptidase can be a bit misleading since the ending is ase, and like the other enzymes present here, you don't want to mess up and think it breaks peptides down. Instead, it breaks down trypsinogen inactive into trypsin active, which goes on to cleave other inactive enzymes like carboxypeptidase, chymotrypsinogen, etc. into their respective active forms. So you might think that enteropeptidase breaks down amino acids, but it only breaks down trypsinogen into trypsin. So trypsinogen is an inactive form, and trypsin is an active form. And we have small intestinal epithelial cell here. We have enterokinase on the tip of these epithelial cells. And these enterokinases, they turn trypsinogen into trypsin, and the trypsin, for example, helps in turning chymotrypsinogen into chymotrypsin, which is the active form, or trypsinogen into trypsin itself. So it's like a positive feedback thing. And we have trypsinogen with the help of enterokinase autoactivation, cathepsin B that turns into trypsin. We have TAP. Trypsin can reinforce itself, so trypsinogen is turned into trypsin, chymotrypsinogen into chymotrypsin pro-elastase into elastase. So these pro ones turn into ones that are active without the pro. We have kallikreinogen, we have kallikrein. So the ogan is removed from the end. So trypsin does all of these things. It's an enzyme itself, right? Pro-carboxypeptidase into carboxypeptidase, pro-phospholipase lipase into phospholipase, and pro-colipase into colipase. So the normal pathway is enterokinase is located in the brush border of the small intestine. And another normal pathway, trypsinogen autoactivation, is a unique feature of human trypsinogen. So only humans have this feature ability that they can auto-activate their own trypsinogen. The abnormal pathway is cathepsin B is located within acinar cells. Trypsin versus chymotrypsin. So trypsin is a digestive enzyme that breaks down proteins in the small intestine secreted by the pancreas as trypsinogen. But chymotrypsin is a digestive enzyme secreted by the pancreas, same thing, and converted into the active form by trypsin. So it is trypsin that activates chymotrypsin. Trypsin is inactive form. Inactive form is trypsinogen. Inactive form of chymotrypsin is chymotrypsinogen. So trypsin is activated by enterokinase, but chymotrypsin is activated by trypsin. Trypsin has alpha beta trypsin are the two subtypes, but and chymotrypsin has A and B. So chymotrypsin has A and B, trypsin has alpha and beta. Trypsin hydrolyzes peptide bonds at the C-terminal site of lysine or arginine, but chymotrypsin hydrolyzes peptide bonds at the C-terminal site of phenylalanine, tryptophan, and tyrosine. So tyrosine and tyrosine is polar, phenylalanine and tryptophan are non-polar, but they are aromatic. On the other side, lysine and arginine are basic. Trypsin is inhibitors. Trypsin's inhibitors are DFP, aproteinin, Ag+, EDTA, benzimidine. But inhibitors for chymotrypsin are hydroxymethylpyroles, boronic acids, coumarin derivatives, peptidyl aldehydes, etc. Trypsin is used in tissue dissociation, cell harvesting, mitochondrial isolation, in vitro protein studies, etc. But chymotrypsin is used in sequence analysis, peptide synthesis, peptide mapping, peptide fingerprinting, etc. That was a comprehensive lecture on trypsin and trypsinogen. What enzyme catalyzes the hydrolysis of peptide bonds, which break down proteins into smaller peptides? Should be aminopeptidase. Trypsin catalyzes the hydrolysis of peptide bonds. So guys, I'll have to go home and do the rest for later. So for now, ciao.